So next up, we've got some really exciting scientists that are going to talk to us about all of these wonderful substances that we are coalesced around today. We're going to be talking about new studies in mycology and fungus-based pharmacology. We're going to bring up the one and only Dr. Sue Sisley, who's a principal investigator at Scottsdale Research Institute. Next up, we've got a founder and COO of ParaBio, Leonard Lerer. And last, we've got Ryan Moss, who's with us from Above Ground Services, a licensed facilitator. Oh, I'm sorry. Philip, welcome. So we're going to start today with Sue's presentation. Sue, welcome.
Okay. Oh, wow. Efficient. All right, I'm going to fly through this. We got just a little bit of time and a lot to cover. So how did we get interested in studying natural plants as medicine instead of just single molecules? I tell you, it really started with my experience with Marinol as a physician in the internal medicine clinic. We were taking care of tons of patients on chemotherapy, and the drug reps would insist that Marinol is exactly the same as cannabis, performs the same. And then when I started prescribing it, people would come back and say, oh my God, I felt totally psychotic, I was paranoid, I had panic attacks, and inevitably they would end up seeking out natural cannabis flower and reporting back in clinic that they were having much better experiences, not just with the nausea, but just overall wellness, mood, sleep. So at that point, I got to team up with MAPS and became the PI on their cannabis trials because I became really interested in the potential of cannabis drug development, especially inhaled cannabis. And then suddenly the government study drug arrives at our lab, and I was so disappointed to see this, anemic material here full of stems, sticks, and leaves, and I was wondering how this could possibly function similar to real-world cannabis. And we ended up completing this trial and publishing the data. And no surprise to you guys that the data, it was a negative study. The cannabis from the government did not outperform placebo sufficiently to be considered positive. But simultaneously, our colleagues were running another trial looking at cannabis for treating PTSD, but they were doing an observational study using this gorgeous real-world flower full of, the sticky resinous material loaded with trichomes. And I always dreamed that someday we would be able to use that material in a clinical trial instead of the material that we were getting. And then eventually we kind of migrated into seeking permission for psilocybin mushrooms. And this is some weird, wonderful fruiting bodies that we were cultivating in our lab. And it kind of crystallized my determination to let these mushrooms grow out to their full expression and allow these guys to be used in studies without forcing them to be over-processed to meet some absurd GMP criteria that FDA has established for synthetics. So many of you know that we ended up using this cannabis argument to sue in federal court. So we spent years attempting to persuade the government to give us our own cultivation licenses for both cannabis, which we did eventually break through. This was a 53-year-old government monopoly at University of Mississippi that was producing this cannabis. But we also parlayed that into getting permission to grow our own psilocybin mushrooms for research. And you can see the study design that we've selected coming up and the timeline I'll tell you about. How we decided on the study design all really emanated from our work with the veteran community. We have really deep ties with all sorts of groups, and I've been learning from them. It started in cannabis. I was very skeptical when they would tell me, claim that they were getting benefit from these plants, and mushrooms and roots, and but eventually I was persuaded that this deserved to be studied, and I couldn't ignore the fact that these veterans who were using this material were incredibly lively. They had a will to live and to be connected to their families and work, and so I jumped on board. We started growing the mushrooms at our lab, and you can see here that this is very different, right, than those psilocybin mushrooms that you may have seen in the real world. And what do you think is special about these? That these are federally legal mushrooms. So I think this is such a silly situation that these are suddenly classified as superior to the other mushrooms and should be used in the only things that should be used in clinical trials. But we're navigating this process. Here we are at the DEA supply chain conference this year. We were amongst all of the big pharma companies, Purdue, Merck, Pfizer, all of them sitting with us, talking about their opioid, the control of opioids, while we're just talking about wanting to grow some plants and stuff. So how, here's what our design looks like, just so you can get a glimpse of. So we're gonna do a phase 1/2 trial. We're gonna study safety, but also add a bunch of efficacy measures. And this is gonna be a gold standard randomized controlled trial. But it is gonna be using real, whole intact psilocybin fruiting bodies, which we're super excited about. We wrote this bill last year and ran it through the Arizona legislature to allocate $5 million, first ever government money, to study real-world natural psilocybin mushrooms. It passed. It is getting ready to be allocated by February of this coming year. Yeah. Thank you so much. Yeah, we're excited, and we've narrowed down our strain now, so this is one of my favorites. And I'll tell you, we're growing all sorts of strains in the lab, right? We got, here's the 222 form that gives me permission to ship mushrooms across state lines, and you can see all the different, B+, Blue Meanies, all kind of classics. And, but ultimately, I decided to settle on Jedi Mindfuck because I enjoy writing that on the DEA forms. Yes, I love that. You can see the Department of Justice watermark on the back. I'm looking forward to their reaction. So what we're going to be doing is exposing same subject to 2 different doses of fairly high dose whole mushrooms. So initially they'll get the 3-gram dose, which is approximately, it looks like it's ending up, our testing is showing us about 30 milligrams psilocybin. Then there's a 1-month washout, and they come back and get the larger 5-gram dose, which will be about 50 milligrams of psilocybin. And you can see here that one of the things we incorporated into our study design is that we wanted an altar. We have a lot of tribes in Arizona that have been consulting with us on how to better communicate with our mushrooms and help them to thrive. And so this is one altar that we have in front of our farm where we bring objects that are meaningful to us, and it's really helped us to have a better sense of how to interact with these mushrooms and honor their consciousness. But the other thing is we built another altar in the front of under the same roof. We have a DEA Schedule I license for the researcher part. So we can do the clinical trials in the same building that we're farming the mushrooms. So what we want to do in the study is have patients come back to the farm, interact with the mushrooms, touch, massage them, feel connected, and appreciate that you're getting a piece of nature here. This isn't a synthetic molecule in a capsule. So here's another aspect of our lab. I'll just fly through this quickly because This is the analytical testing portion, which we're also really proud of because we've been working on validating methods for potency testing in whole mushrooms, which we're getting ready to publish, keep it all open source. This is, we're looking at the difference between potency in the stems and the caps, and we grind it down in our cryo mill so we don't convert too much of the psilocybin into psilocin. So we're grinding it with cold temperature, and experimenting with different solvents to see which one gives us the most optimal and accurate potency testing. And this is our HPLC machine. Many of you guys have seen this in the chromatograph showing your psilocybin and psilocin peaks, but also tons of other peaks that you see on these that we haven't identified yet. So very curious how all these molecules may be interacting together. So what would happen if we had more resources and less regulatory hurdles? Our dream is to make federal right to try law work to protect Schedule I drugs. So we're on a mission to prove we have to complete this FDA phase 1 trial. That's what the law says. And then we are allowed to respond to patients who are asking us already. We're getting flooded with inquiries from people who want this medicine. And so our hope is that we can eventually be able to gift these mushrooms to patients all over the country after we meet criteria for federal right to try law. And the other dream is, this is our second lab in Montana overlooking Flathead Lake. It's also an American Legion post now. And what we're doing, we're hosting ceremonies for veterans there, and we're eventually, we have a DEA Schedule I license at this lab also to be able to do clinical trials here. So we're excited about the opportunity to have journeys outdoors under the big sky. And so really our dream would be to be able to encourage people to feel that connection, be able to hold newly harvested mushrooms, feel that connection to nature. And you can see here, so the first step is finish that phase 1 trial so that we can be defined as an investigational drug by FDA. This allows us to then meet criteria for right to try, and the DEA has said that we need to create a network of Schedule I license holders around the country. So if you're a doctor out there, if you have a MD, DO, PhD, and you want to be a recipient of these mushrooms eventually after we complete phase 1, please come talk to me after, because we're developing our list and we'll be able to help you get your Schedule I license through this process. And overall, our work is trying to deepen our relationships with these groups. The veterans, we had an incredibly shocking experience this year with American Legion, where we got our little American Legion post in Montana got the first ever psychedelics resolution passed by the hyper-conservative American Legion. And so we're working with all these other VSOs now, the firefighter associations, to help them stand shoulder to shoulder with us as we push the government to move forward. And I just want to invite you, this is our language from our state bill. We would love to see this become model legislation in every state. We want all of you to go after your own 5 million, 10 million out of your state government. So please come up after and talk to me, and we'll all work on this dream together of getting rid, using research to get rid of the criminal penalties around these gorgeous creatures, and hopefully dream of a time when everybody can grow these at home and enjoy the therapeutic benefits of farming their own medicine. Thank you very much.
Captivating as always, Sue. Thank you so much for the heroic work that you've been doing for a very, very, very long time in this space. We all owe you a debt of gratitude. Thank you. Next up, we're going to receive a complimentary perspective from Leonard Lerer.
This is my first visit to Portland, so I paid a pilgrimage, obviously, to Powell's bookstore. Obviously, I fondled the Burroughs and the Ginsbergs and all that, but I found a fascinating book about the medical treatment of elephants. Obviously, being a physician, I turned immediately to the parts about. Firstly, the book wasn't very big, because there's not much known about the medical treatment of elephants. But I turned to the section which is about surgery of elephants, and it said there that you needed you had to have special instruments made in order to do surgery on elephants. And it kind of got me thinking about the whole idea of the elephant in the room. And we spent about more than 3 years now dealing with the elephants in the room in psychedelic medicine and psychedelic science. Why did we get there? What is the current context? What have we done? Okay, what have we learned? And what are the implications of what we've learned for drug development? That's the next one. So when we started the journey, we were fascinated. In order to develop a drug, we really had to understand, this thing is being used. Up to 4% of Americans are using psychedelics. And I don't want to go into this in great length. It's published in Frontiers in Psychiatry. But I just want to make the point is that, yes, people are using psychedelics. What we found is that those who were using it for the areas which we were interested in, which was major depressive disorder, OCD, okay, they were young, they were well educated, and a lot of them were not getting better, okay. In fact, a substantial proportion of them were landing up in the emergency rooms of hospitals with their original condition or the even original condition exacerbated. The next piece of work I want to show you is one which was presented at the American College of Neuropsychopharmacology last year. And we haven't published it this year yet because we've had substantial problem with the variability of the flushes between the mushrooms. And essentially what we've done here is we've looked at done a survey, and at the same time, we've analyzed the minor tryptamines or the tryptamines of the psilocybin biosynthetic chain and seen, could we correlate the subjective effects of mushrooms with the proportions of the various tryptamines? And fundamentally, the answer was no, not really. And I think we could debate this matter ad nauseum. I just want to show you this, and we're more than happy to share our raw data. We're redoing the work now with Alan Rockefeller and Felix Bley, and we should have something out within the next while. The most important thing I really want to show you is that I think from a preclinical point of view, we have laid to rest the debate over the efficacy of natural psilocybin, in other words, full spectrum mushroom extract, versus chemically synthesized psilocybin. From a effect point of view, in other words, in disease models of depression and OCD, from a neuroplasticity point of view, from an early immediate gene expression point of view, and from a metabolomics point of view. In fact, I think that all that needs to be done now is that we need to gird our loins and go out there and do a clinical trial which would be the efficacy of full spectrum mushroom extract versus chemically synthesized psilocybin. And this paper will be published in molecular psychiatry in the next couple of weeks, and is being presented next week. We're presenting it at American College of Neuropsychopharmacology in Tampa. So fundamentally, what are the elephants and how are we dealing with them? The first elephant is undoubtedly the elephant of access, okay? That is that it is most unlikely that we can treat the millions of people who would benefit from entheogens and psychedelics if we have the psychedelic experience or the trip. And we are devoting a considerable amount of research to this. The next question is that of efficacy. We want the most efficacious entheogenic drugs available, be those chemically synthesized, be those produced through metabolic engineering. or maybe they will be produced through some or other natural method. And our work there extends from we're the first in the world to produce 5-MeO-DMT from the immortalized cells of Encelius alvarius, the Sonoran desert toad. And I'm sad to announce that one of our toads who donated part of her parotid gland, after living with us for more than 3 years, passed away. Betty passed away a few days ago, and a source of great sadness to us. So I won't ask for a minute of silence, but I just want to acknowledge Betty, the toad who was the first source of immortalized Encelius alvarius cells. And the third challenge is undoubtedly that of availability, or what we would call drug supply. And that's where we are looking very much at new methods such as metabolic engineering to produce sufficient quantities of psilocybin and the other tryptamines and other potentially therapeutic entheogens. So I'll give up the rest of my time for discussion, and thank you very much.
Well, we're sorry to hear about the loss of your colleague. I can say, back in graduate school, I worked a lot with antibodies, and when the rabbit died who was producing the antibodies, it was very disruptive to the work, so I can appreciate it. Next up, we're going to have Ryan Moss, the CEO of Filament Health, to share an experience with us.
Hello, everyone. My name's Ryan Moss, and I'm actually the Chief Science Officer at Filament Health. Filament Health is a natural drug development company focused on developing natural botanical drugs, which is kind of what we've been talking about in this panel. We believe that this is the best way to get these psychedelic medicines into the hands of as many people as fast as possible. We are firm believers in natural products and the power of psychedelic medicine. We believe that the current state of drug development, focusing on synthetic small molecules, potentially leaves a lot on the table through the entourage effect. And so we believe that the best way to test whether this entourage effect actually exists is to perform clinical studies in humans. And this is kind of where our research question began, is how do we get these botanical drug substances into FDA-approved human clinical trials? The first challenge that we needed to overcome was getting the drug into an actual form that would be amenable to a clinical study. Interestingly enough, the FDA in 2015 published the Botanical Drug Guidance. This guidance outlines the expectations and circumstances by which a botanical drug could be approved and developed. By their definition, a botanical drug is a complex mixture of components with a substantial prior human use. It must be standardized and consistent. We believe psilocybin, through the consumption of magic mushrooms, fits this bill exactly. So the initial drug substance and raw material that we started working with was magic mushrooms. After developing all of the appropriate testing methodologies and validated testing methods, we procured a federal license in Canada to actually manufacture and ship the product around the world. And then we identified the goal of actually wanting to get into the clinic as fast as possible. So over the past 3 years, we've actually cultivated thousands of pounds of magic mushrooms. We've cultivated well over 100 different strains from over 10 different species of psilocybin-containing mushrooms. We've performed experiments on different styles of growing, different substrates, different temperatures, different drying methods, all to try and achieve a consistent raw material from which to make a botanical drug. One hurdle that presented itself while working with magic mushrooms is that they are far too variable as the raw substance. An individual mushroom can vary in psilocybin content by up to 5-fold. So if you were to give 2 mushrooms from the exact same flush to 2 different people, you would actually have very little certainty in how much active substance you are giving that person. And that's kind of not really amenable to a clinical trial where you're trying to test the efficacy of the drug. So if you're giving variable amounts to every single person, it's going to be very confounding in the results. One could imagine finding a solution to this by taking a big lot of mushrooms, grinding them all up, and getting them tested and assigning a psilocybin content to them. But this actually doesn't really fully solve the problem, because then the next batch, you're actually going to be giving a different amount of substance to the person. So this person in this part of the clinical trial is going to get 5 grams or 3 grams, and then the next part of the clinical trial, they might have to get 6, 7, 1, 2. It's far too variable for a clinical trial setting where you're actually trying to prove efficacy. So because of this inherent problem in natural products, and it's not just magic mushrooms, this problem is inherent in all natural products, we developed a series of extraction procedures to make a whole extract, very carefully developing and designing this process to retain the full chemical profile of the secondary metabolites of the raw mushroom and leaving that intact in the final extract. This both allowed us to study the effect of the full botanical profile, but it also allowed us to take any variable mushroom that comes from our chosen production strain and get the exact same product out the other side every single time. Using this developed technology, we were able to develop 3 different initial botanical drug candidates based on Psilocybe cubensis mushrooms, 2 of which have unique chemical profiles which are different from each other, and 1 is actually a different dosage form which uses a different route of administration. Each of these products are standardized to an exact composition and content of all of the tryptamine alkaloids that you may have heard about. Each one of these products has shown shelf stability for over 2 years' time, and each one is contained in a size 2 capsule, so people wouldn't need to be consuming powder, which, I mean, some of us enjoy to eat the mushrooms and taste them and feel them, but there's lots of people in this world who are in dire circumstances who can't take mouthfuls of powder and need something that's a small enough dosage form that is amenable to their situation. So with these products, with these 3 initial products, in October 2021, our first clinical trial at UCSF was approved by the FDA. It uses all 3 of our mushroom product candidates, and it contains all of the natural tryptamine alkaloids that you may have heard about, so psilocybin, psilocin, baeocystin, norbaeocystin, aruginacin, norpsilocin, 4-hydroxytryptamine. It contains all of those things in each one of these botanical drug products. This study was actually the first ever FDA-approved study with using a natural psychedelic rather than an isolated or synthesized small molecule. This study is primarily assessing the mystical experience of each participant using each of the drug candidates. It is a randomized, double-blind, 4-arm crossover design, so every single person is getting every single one of the drug products. And the study is set to be completed by Q3 of 2024. This should shed more light about the entourage effect and how these different metabolite profiles actually affect the therapeutic potential and mystical experience. Due to the relative difficulty in procuring GMP psychedelic substances for academic researchers, since we had success going to the FDA and allowing these to go into clinical trials, we've actually been very active in providing these drug substances to other academic researchers at no cost. Over the last 2 years, since October 2021, we've actually now been approved, our drug candidates have been approved for over 30 clinical trials around the world. So researchers are interested in using these natural substances. This includes many different jurisdictions, the USA, Canada, the UK, EU, and even Israel. So in addition to providing these drugs to other researchers who are interested in using a natural substance, we are also very active in the SAP program that's in Canada. It's the Special Access Program, which actually allows doctors to prescribe experimental medicine to people in extraordinary circumstances where absolutely nothing else has worked. And luckily, Canada seems to be very proactive in this approach. We've been approved to ship over 300 doses of these products to very sick people. And these are mainly for the indications of end-of-life distress and major depressive disorder. When we began this work, most colleagues would tell us that it simply wasn't possible to get the FDA to agree to use a natural substance when there was small synthetic molecules available for the same purpose. But given the results we've achieved this far with our magic mushroom-based products, we are continuing to do this work with other potential botanical drugs. So, in conclusion, we're very proud of the research we've done this far. We believe that the tide is turning and that the complex botanical mixtures will become more prevalent in the scientific and pharmaceutical drug development spaces going forward. Thank you for your time.
Thank you so much, Ryan. One of the things that I was the most excited to hear about in your talk was the shelf stability stuff. Sue and I have talked about this before. It's just a total unknown. You know, how long do these products stay as potent as they are? What specifically is happening to them over time? I'd love it if you all could describe this shelf stability and how do we stabilize things, and what are you observing? How do things change over time?
I mean, there's lots of factors in shelf stability of drugs, all the way up to how you actually store them. You need to be very careful and very specific in exactly how you store, even up to whether a bottle is horizontal or vertical might actually affect how these drugs are stable. But magic mushrooms themselves, even when you dry them as much as you possibly can, still retain some moisture, and that moisture leads to enzymatic degradation, and mushrooms contain enzymes that actually degrade psilocybin itself. And so that's a huge problem. You also, in this whole substance, you have a lot of polyphenols and things like that that can degrade over time due to light or temperature or water contamination and things like that. So I think just being very careful and having that development in mind when you're trying to construct this product.
Mm-hmm. So what I'm hearing from you is we've got moisture as a concern, we have temperature and light contamination or light-induced, photo-induced changes. You think those are the three things that are primarily contributing?
Sure, and then you compound those by, if you're grinding up the mushrooms, you're getting a lot of surface area, so all of those things become compounded because there's just so much surface area for those things to act.
I see. So the more stable long-term solution, if as long as it's not an extract, would be the whole mushroom compared to like a ground product?
The whole mushroom, and like when you think that it's dry, it needs to be drier than that.
Okay, great advice. Sue, how does that compare with your observations?
Yeah, sure. I really admire Ryan's, his pathway and what they're trying to do to organize a pharmaceutical grade product that is reliably dosed and has a reliable shelf life. In our case, we're discovering, as many of you have, that, once this does convert to psilocin, it's a very unstable molecule, and yet I don't want to combine tons of preservatives to make that molecule linger. I'd rather have to study the real natural mushroom in its form, in what Mother Nature intended, and not end up infiltrating it with tons of other material that make I think that the public should have access to both pathways. Like Ryan said, there are a lot of people that will never want natural mushrooms, and then there's a huge faction of the public that does want it, and these mushrooms are doing their best job to defy standardization. Like he said, they are working hard to make sure that they are not pigeonholed into some FDA paradigm of how they should behave. And personally, I'm rooting for the mushrooms. I want them to keep going.
And what about batch-to-batch variability? In the cannabis space, this has been probably the most challenging component to having reliable, consistent medicine for people, is that this plant is wild. It is extremely sensitive to its environmental conditions. So I'm curious, in the the strains that you're growing. Strain is actually the right word in mushrooms. It's not in cannabis, by the way. It's varieties, dang it. But these strains that you're growing, how stable are they between harvests, or how sensitive are they to the environment?
They're incredibly sensitive to the environment. In our facility, everything is controlled down to the T to try and get, like I said, initially our goal was to just create something that fit the definition of a standardized and consistent thing that we could dose and give in clinical trials. And even with the goal of doing that and having our laboratory conditions amenable to being able to control every little parameter, these are living organisms, and they, very much like what Sue just said, they're trying their hardest to not fit into a specific mold. And our solution to that was obviously extraction, but I admire Sue and how she's going to solve that issue.
It's good.
I really don't want to enter into this controversy. All I will say, because I have a very different point of view on this, but anyway, all I will say is one thing that nobody's really mentioned is the issue of genetic stability. And here one needs to look at the work of Jason Slott and Felix Bley and Dirk Hofmeister. Not only do you have the issue that the actual physical growing conditions are different, and we were one of the largest growers of psychedelic mushrooms in the US until about a year ago. Okay, but you also have the question of genetic drift, okay, even within a flush of mushrooms. And I cannot see how you'll even get close to some kind of standardized, never mind FDA approvable, forget about that, standardized mushroom extract unless you have the phylogenetics on every single grow of mushrooms, and that's not being done.
Yeah, that's a great point.
That's true.
And especially, the life cycle of these creatures is so short. So, the number of generations or potential, flushes that you have within, a fiscal year is like pretty substantial compared to, other natural products where, a lot of, plant-derived medicines are seasonal. There's one harvest per season. So that genetic drift, within this space is a lot different than it would be in others.
And if I could just make one other point, it's not only that the psilocybin levels vary within a mushroom grow. The relative proportions of the minor tryptamines, aerogenicin, baeocystin, norbaeocystin, etc., the relative proportions also vary substantially. That's why we could never publish the work, where we did the naturalistic survey, experience versus how much. Okay, we did see something. For example, higher levels of baeocystin were associated with greater body load, etc. But none of this was anywhere near the rigor, because we found that we did two sets of mushrooms, okay, and we found there was a huge difference in the relative proportions of the minor tryptamines in each of these groups, which became a statistical nightmare for us, But I will say that, it seems that the relative proportions do have some effect on how people experience it from a body load, etc. perspective.
That's a good segue to another question that we had, which was that, these sort of entourage effects, or the difference between the effects of, isolated psilocybin versus these full spectrum extracts or whole fungus, quantifying those differences inherently asked you to focus on, okay, well, what questions are we asking? What endpoints are we measuring? So, for example, we might say, okay, is depression symptoms our endpoint? Is tolerability or side effects our endpoint? Is the duration of the experience or the intensity of the experience? So what do you think about in terms of, okay, we're all working toward this goal of doing this head-to-head examination of, purified psilocybin versus these whole products. What endpoints are you thinking about?
No, I think that's pretty clear. That's what we have 2 years of preclinical work on this. In a mouse model of depression, in a zebrafish model of OCD, does the one work better than the other? The answer is yes. In early immediate gene expression, does the one work better than the other? Yes. In all the markers of neuroplasticity, BDNF, etc., does the one work better than the other? Yes. in all the metabolic markers which are associated with neuroplasticity. Is full spectrum mushroom extract showing more of these neuroplastogens? The answer is yes. I think we've shown that. The question is, does that have any implications for clinical development or not? There we're not sure. Okay, that's the next question. But as far as we're concerned, Compass Pathways is not quaking in the boots. It's boots because of what I'm saying. Okay, maybe it should be. Right. Okay, but the evidence is there that a mushroom, that full spectrum mushroom extract, never mind eating mushrooms, just full spectrum mushroom extract, seems to be a more potent neuroplastogen and seems to have, actually it does have, in our OCD model, which we'll present next week, it has a stronger therapeutic effect in a very, very well-validated set of preclinical models. And I'm not saying this has any implications here, but it's nice to know,
Yeah. Well, and also, I think proof that they maybe are concerned about your findings is that we've asked Compass and USANA to provide synthetic material so we could do a head-to-head trial finally, looking at synthetic versus whole mushroom, and they refused to provide the drug. So there must be some type of worry that, and their best way of handling this is just to keep impeding FDA drug development process, which is what we're dealing with now. I mean, that's the reason you see, when I was talking earlier about how we're only doing phase 1 and going straight to the public, is because that's why we pushed for right to try law in this country and got it signed in 2018, because we knew that FDA would never embrace whole mushroom and allow it to move through phase 2 and phase 3. With phase 1 trials, FDA doesn't mandate GMP. That's a little known fact, that you don't have to meet these overzealous GMP criteria for phase. So we can do phase 1 with whole mushroom, but then after that, we would have to meet all the same rigorous, harsh criteria that Ryan's having to deal with. But that's why I'm so envious of his path, because he's working with precision dosing, and we're the opposite. We're working with dosing ranges and trying to persuade FDA that is still safe and it's still ethical to do these trials.
I'm not sure if I share your paranoia about USANA. Well, I don't share your paranoia about USANA because we're in Chicago and they're right next door to us. They're very big and very powerful. So.
Yeah.
But I don't think I should, because what we have to understand is, full spectrum mushroom extract, natural mushrooms are better. That's not a drug. A drug is, there's still a journey which we're on, which is a tough and expensive journey to find out why. Is it the minor tryptamines? Somebody this morning said, it's the harmines and harmalines. That's nonsense. They're in such tiny amounts that they cannot have any effect. Which one of those tryptamines? Is it the aerogenesin? Is it the baeocystin? Maybe it's something else in that extract. This is a very, very tedious job and a very, very expensive job, which involves a huge amount of organic chemistry, etc. But we're doing it slowly but surely. But to us, the discovery that full mushroom extract is more neuroplastic and more therapeutic in preclinical work than chemically synthesized psilocybin is only, it's the end of the beginning. It's not the beginning of the end of chemically synthesized psilocybin.
Well, the other thing though is FDA doesn't require that when you're doing drug development, you don't have to prove the mechanism of action. Right. So if whole mushrooms prove to perform better in clinical trials, the FDA isn't going to mandate that we sort out which tryptamines or alkaloids are actually exerting clinical effects. So that's very helpful for us, because we don't want to go down this path like Len is going down, because we don't have the time. We want to get medicine to people right away, federally legal medicine.
Listen, I violently disagree with you.
Yeah.
Okay? And I'll tell you why. Because I believe that we are on the path to get medicines to people. Okay? Because there is no way that millions of people are going to be able to be treated with mushroom with stuff which is grown. Okay? There's no way. It's not going to work. There's already a supply crunch. of this. There's no way. The only way is if we develop a drug that can be prescribed by a physician, okay? And to me, it's not an ideological argument, okay? It's not an ideological matter. It's just a plain practical fact, is that people with major depressive disorder, addiction, OCD, and a whole swathe of other conditions should get access to entheogen-based therapeutics as soon as possible.
Right. Yeah. But I'm a hospice doctor, so I need people to get it urgently. I don't have time to wait for FDA to finally embrace this. That's all I'm saying.
I have no problem with that. And I'll supply you with chemical psilocybins. DEA license to DEA license.
I think I would have to push back on the fact that mushrooms can't supply all of this medicine.
Yeah.
You can look in the food industry, the food ingredient industry, everywhere where these are grown on massive, massive scales and hundreds of millions of people consume them. So I think that it is viable as a scalable business to use it as a precursor. In fact, there are many, many drugs that are produced from even animal waste byproducts and plants like the yew tree that are viable drugs that are being given to millions and millions of people.
You know what? It's an academic debate. Because
Sure.
as soon as the market for psilocybin grows big enough, the Chinese, Italian, or Indian API manufacturers, okay, will eventually set the same reactors that they have to make other chemical drugs. They'll set that onto psilocybin, okay, and we will be flooded with psilocybin.
In the small molecule psilocybin, I totally agree. But drug products made either as whole mushrooms or from whole mushrooms, it's a lot harder for them to replicate.
But there's no doubt about it that psilocybin is therapeutic. Chemically synthesized psilocybin is therapeutic.
Absolutely agree.
And that will be the drug. Okay,
Yeah,
and there will be no shortage of it.
This ties in nicely with one of our audience questions, which is this relationship between psilocybin and psilocin, right? So when we're talking about the active, binding at the receptors in the brain that are initiating these experiences, it's all mediated by psilocin. So, how do you see this balance of, preserving psilocybin or producing psilocybin versus the shifting narrative around quantifying psilocin or trying to stabilize psilocin? What's the relationship there?
Well, in our phase 1 study, we are actually the two different chemical profiles of the two different main drug candidates. One is the dephosphorylated tryptamine heavy, so psilocin and all the various tryptamines being dephosphorylated. And so we should be able to get some data around that question of psilocybin versus psilocin.
That's promising. Well, this has been a really, really exciting conversation, super lively, fantastic.